glycophile somehow, which is probably quite unusual for your company, because I guess probably most of the people normally are in the cheminformatics, more general small molecules. However, I think it's great that they have the opportunity to present our field, the computational design of glycomimetic inhibitors, and particularly carbohydrates. So, as you can see already on the title slide, is an example how carbohydrates are involved in biology and also in medicine. What you see here is a glycolipid interacting with a norovirus. So norovirus recognizes its host by a simple carbohydrate that you can see here, which is a fructose, and is very specific for that fructose. It doesn't recognize a galactose or any other carbohydrate specific to fructose, and that's how it finds its host. So it would be, of course, very interesting to find something that mimics that kind of structure in order to prevent, for example, norovirus infection. So before I continue, I would like to give a few maybe a few words on our company. Our company is a privately owned company and a very small company in Göteborg in Sweden. And our main objective is to provide for smaller and medium-sized companies computational chemistry services. As a software, we use typically now Biosolvit software since 1 year, and also Yasara, Autodoc, and we also have used other software previously. A lot of development is also going on in terms of reporting and analysis, and you can also see some of the customers we served over the last couple of years. Before I joined Biognos, I was in a field called Glycomimetic, and as you can see from that side, there's quite a significant amount of different initiatives going on in the glycomimetic field. And if you're interested in this kind of part of the carbohydrate, computational carbohydrate part, which is more closely related to bioinformatics and not so much to modeling, it's about databases and storing carbohydrate structures in relation from mass spectrometry and so on. There are 2 books dealing with that topic and also a few reviews. One actually came recently out in 2014, which was also a very nice review to give an overview on the field. When people talk about carbohydrates, they usually think as carbohydrates as part of being food or nutrition, or maybe biofuel in terms of renewable energy resources. It's not yet so much into everybody's head that carbohydrates actually play a key role also in medicine. And as you can see here from that overview taken from that review that appeared recently, here are some recent developments in the synthetic carbohydrate-based diagnostics and vaccines and therapeutics. What we talk about today essentially is that part here, carbohydrate-based drugs or glycomimetics. And probably the most well-known example here is Relenza, which is a flu inhibitor drug. But also other areas are equally important, as you can see, diagnostic tools, also carbohydrate-based therapeutics, essentially like glycoproteins and vaccines, carbohydrate antiparasite or cancer vaccines. So if you're interested in that field, here there's an excellent review which you can actually then look into more details. The field of carbohydrate goes back, well, a long time ago essentially, but it started, the structural started essentially in the 1990s. So in 1910 when Fischer before 1910, when Fischer discovered essentially that the structures are hexoses and looked into the structures. The first application of carbohydrates in the clinical use started already in 1930, but it took another about 70 years essentially before the full synthetic heparin reaches the market. It's marketed under the name Arextra. There are other fields, other things emerging, and there's Relenza and Tamiflu approved for flu treatment. This is also keystone in the field, essentially was at the end of the 1990s. There are several drugs actually that contain carbohydrates. Some examples are shown here. So the drugs range from epilepsy, anticoagulant, diabetes, and also antibiotics. So despite in generally it's not so much talked about carbohydrates being drugs, there's already quite a significant selection on carbohydrate-based drugs on the market. Probably the most well-known is Zanamivir, or the market name is Relenza, and that essentially was like the keystone essentially in the design of glycomimetics. This is essentially how it all started. So Mark van Idstein and his group at Monash University in Australia, so they probably did the first rational design of a carbohydrate drug. So they saw essentially here a small pocket in this area which was not filled, and by using the GRID program of Woodford, they found that it would be well suited to accommodate a positively charged group. And so they put it there for guanidino group, which then really made a significant impact on the affinity of that compound towards influenza virus. And nowadays, a follow-up compound that is probably more well-known at the moment is Tamiflu. Everybody probably knows about it. It's the advantage of Tamiflu is that it's a tablet, but the disadvantage might be that it might not be so effective anymore, and so Relenza might be still a very interesting drug to use. The same group recently published a paper and have another success story to report, and it's about now they designed a potent inhibitor against parainfluenzavirus, hemagglutinin neuraminidase. The idea was here, this is actually a very difficult target because of the broad binding site. It's a combined hemagglutinin and neuraminidase, and so the binding site is not as tight as in the neuraminidase or flu neuraminidase case, so it's a bit more open, and that makes it much more difficult to just design a drug. A key observation in that context was by molecular dynamics relations. It was realized that this loop here is flexible, and so the design was targeted towards an opening cavity that you can see a little bit here in the crystal structure, and by extension with some triazole-linked phenyl and some ether compound here, they could significantly improve the affinity by essentially from the normal, basic and natural kind of inhibitor, that the 5-actoine, down to essentially almost 1 micromolar. That's a very exciting story, and here you can also see now how CSAR could be used essentially in order to look into these kind of compounds. What you see, so the green, for those people who are not familiar with CSAR and height, the green balls essentially give graphical representation of the height score. When it's green, then it's a favorable. The sum of dissolution and interaction part is favorable. That means it's a good atom essentially in terms of interaction. If the atom is red, then it might actually, in most of the time, is an unfavorable dissolution term, and which is not counterbalanced by any interaction with the protein. So here in this example, there's this oxygen of the ring oxygen, which is red, but this is in the case for most carbohydrates actually, and so there's not much that one can do here, and because very often this is a key, sometimes this is a key oxygen here. But it looks pretty good. So essentially, it's all green. So this Zanamivir, although it's only 250 micromolar inhibitor for the for the parainfluenzavirus, hemagglutinin neuraminidase, it actually shows good scores here in the CSAR display. The new version of CSAR has the possibility also to do docking. So for that webinar, I tested a redocking of the ligand, and the good news is essentially the height scored version is the best one. That is the closest to the crystal structure, but there are many structures that are actually not even close to the crystal structures, and they also get a bad score. So the scoring works. but the docking is maybe not completely efficient at the moment. But it's also very interesting that the scoring of the crystal structure gives a very good prediction of the affinity range that is actually the experimental affinity range. So in principle, scoring of crystal structures with height works very well. And the docking is in CSI anyway pretty newly implemented, so it might be possible to do a little bit more development here, so it's more reliable. But it's a very, very good. Sometimes we get very good results, and so in this case, I just wanted to show you that you not always trust essentially what you get. What is much more robust in that context, and also in that example worked essentially perfect, was the redocking of the ligand with FlexX. So we get here RMSD value of 0.36, which is essentially perfect, which is practically identical as the crystal structure. How was that achieved? That was actually achieved by having the solvents that are part of the binding site included in the docking. And so if we now, so what I do now essentially is I made a little test project. What I did was I used the crystal structure that actually has had Zanamivir in it, and I prepared with the LeadIT interface, which is a very nice protein preparation wizard, a version that actually had no waters in the docking, and also one that actually had waters. And there is also there are many crystal structures available of para-influenza hemagglutinin neuraminidase. So I picked one that is an APO structure that has no ligand in it. So I was interested, how does the selection of the waters, yes or no, and also how does the selection of the receptor influence the result of the docking. So in each of these four receptors, the compounds from the paper were actually then docked. What you can see on the left side is the FlexX score, and with the four receptors you can see when the APO structure gives a much worse score, so it's not so good shaped, obviously, for accommodating the ligands, and also the dry version of the receptor with the Zanamivir in it is actually, it gives a much lower score, much better scores essentially than is achieved when the water is included, and also so also the sequences of the compounds is much more closer to experimental values. When actually then all the poses are then rescored with the height, which is the height scorer, which is an extra software in the LeadIT package, then you can actually see first of all, there's a little bit more diversity now in the scores. So they're not all like clustered together. And also here, particularly in the water case, there is now excellent agreement with the ranking order. Not every compound is actually perfect in the ranking, but overall it's a clear sign that the ranking has improved. And so what we see now essentially also that the little blue X, which is a new 5X, that should be always the weakest one, and this is very, very nicely here, but it's also very nicely here in the FlexX. And also the ones, for example, the C10 and the green one, the C9, which is this one here, the blue one up here, that it has now an excellent score. The bigger ones do not have such a good score, and the reason for that is this receptor is not shaped actually for accommodating the ligand. So the cavity that was actually targeted with that kind of receptor is not very well formed. So one probably would need then to use another receptor structure where that cavity actually is formed. In the publication, they also see that by running molecular dynamic simulation and then getting probably a snapshot out, if I understood correctly, and minimize it probably, and then docked into that snapshot. So the scores is one thing. The other thing is actually are then the ligands actually docked correctly? Are the poses correct? Because essentially you can score everything, but if the pose is not correct, your score is essentially meaningless. So what we see here is this is the way how I do it. With the software that I use is called a confirmation analysis tool, in short CAT. So what I can do there is I define a couple of points in space. They call it hotspots in the CAT terminology. And for that I can calculate an atom site occupancy score, which essentially gives me a measure for how well essentially these ligands dock into a certain space. This is particularly useful because in typically when you're docking diverse compounds, and RMSD is not very good because you don't have the matching atoms typically, but in order, if you can check essentially if a certain pattern in space is occupied by the right type of atom, then you can calculate a score, which is done here. So overall it should be 11 because there are 11 spots here. So you can also see that in the water case, essentially there's a much higher percentage of the compounds that actually have the correct score in terms of volume score. The second observation is that the blue one is here the one that is the outlier, and this is understandable because new-5-actoine, which is a chair-type molecule, doesn't fit very well into this kind of scheme, and then you can see essentially the method works, so you can clearly discriminate here between the post quality. So there's a difference between post quality and score quality, like energy score quality. So the first you need to get the post right, and in carbohydrates there's very often known what is right, because there's a natural carbohydrate that typically sits in the binding site, and you want to mimic with your compound that kind of interactions, and then plus having some substituents that increase affinity, then actually get a better inhibition. So what is the take-home message essentially from that exercise? Is essentially that the docking outcome is significantly influenced by the receptor that you pick, and as you can see on the left-hand side here, the receptors are actually well, they are not identical, but I would say they are in the range of typical variations of crystal structures. And so essentially, depending which crystal structure you pick for your docking, you can have completely different outcomes. And the second is, well, water makes a huge difference in the accuracy of the docking. And this is also understandable because if you actually display the waters that are in the crystal, as you can see on the right-hand side, a significant part of the ligand is actually swimming or embedded in water and has bridging waters. So it's quite straightforward that actually the waters may play a key role here in the binding of this ligand, which is also very often neglected in docking experiments, and I think Biosolvit has offers here some good tools in terms of including the water into the binding site. Now I've mentioned CAT. CAT is I developed CAT since essentially like 15 years now. Originally this was aimed at giving me a tool for high throughput analysis of molecular dynamics simulation. So, and then essentially the data should go automatically essentially into a format that allows me to interpretation, or sometimes even go into databases. And so one example is shown here. This is when I calculated about, let's say, 1,000 MD simulations of different types of carbohydrates. There are conformational preferences and the glycosidic linkage, which is like a Ramachandran type of representation of the linkage. And so Thomas Lüdecke then put this data onto the web, and now crystallographers can use that for quality assessment of the crystal structures. And as you can see here from the left-hand side, the dark spots are crystallographically derived structures from the PDB, and the background is then the low energy area where this kind of linkage that we have here, which is in this case a MAN beta 1,4 glucanac linkage, which is essentially present in every N-glycan that is on more than 50% of the proteins, and people are sometimes not aware that most of the proteins are actually glycosylated, so obviously glycans play also a very key role in some cases, at least in the glycoprotein world. So we can see essentially that many of these linkages fall into a very well-defined area. There are some outliers, and this usually is very important to check essentially whether this is a real outlier, scientific valid outlier, or whether this is just an artifact in the crystal structure, or the crystallographer just didn't pay enough attention to refine that pose. So and this is how CAT works in principle, because it's aimed at high throughput analysis of a variety of data types, particularly MD simulation, but also glycomimetic, bioinformatics, and simply data processing. So this is a XML type language essentially. So you read a template structure, for example, to define what you want to measure. You analyze then your archive, a trajectory, for example, or other SD file or whatever, and then you save essentially your output. And what is also possible here, you can generate nice plots. A few of them you have seen already, and then you just run it in a Unix script essentially, and then usually it doesn't take very long to perform even complex analysis. So in terms of high throughput processing, we now go now to a structure-based virtual screening example. The Relenza, the neuraminidase actually is one of the few examples actually that also made it into the chemoinformatics field. So in the so-called DAT dataset, this is a directory of useful decoys. There's also a neuraminidase target in there, and the decoys essentially. So essentially, the virtual screening test that we do here aims at the question: can we, if we mix active compounds into a set of other molecules, can we actually derive back essentially our active compounds? And does the talking software or the screening outcome tell us, okay, these compounds are active, or most likely active molecules? And what you do here, essentially, you take 49 active molecules, you mix them with decoys, which have similar physical chemical properties, and then the question is now, can the docking software essentially get the actives higher in a higher rank, a higher affinity score than the ones that are actually not binding? And so the diagonal here represents essentially the success rate when you pick randomly compounds. Essentially, if you have half of the library tested, then you probably have 50% of the actives found. That is rather straightforward. The ideal case, of course, would be essentially if you have 49 tested, then you have all of them found. That's very idealistic. That never happens. So what we really get now in this essentially is the red curve. So if we go like 100 compounds, if we test 100 compounds, we get about 35% of the actives in our assay. And so this is now really important when you go into kind of de novo search for small molecules. The better the docking software can deal with the target, the more this curve is into the ideal case, and of course the more money you save essentially, because that saves you from testing a lot of compounds that are false positives. So that, although this is already works here in the neuraminidase case, is still not too exciting, because essentially you still need to test about 600 compounds out of 1,600 in order to get all the actives. So, but this has to do with the situation that we actually have very similar compounds, which is typically not the case in a real small molecule library. For that reason, I mixed about 8,000 other compounds into the set, and then we get a much, well, the library is much bigger, so the enrichment is the same, but here the percentage or the situation looks quite different. And the reason for that is shown here. So we have the blue ones, which is our actives, the green ones, which are our decoys, and the gray ones that are our other molecules, and they are not related at all to any of these. And so they typically have generally a much unfavorable score. So in this case, even if you have a larger library with the same enrichment, you can actually be quite successful in fishing out compounds that are active in a virtual screen. We actually use that kind of approach for finding active compounds against a carbohydrate target. Israel Ladavsky, Professor Israel Ladavsky from Tel Aviv in Israel, asked us to screen e-molecules database of 5.2 million commercially available compounds against his freshly crystallized heparinase. So we did that, and so we did that with GLIDE in those days. So with high throughput version, we did a pre-screening, and the top 1.2 million were then essentially with single precision GLIDE screened again. And then from the top ones, essentially we did some filtering for drug-like molecules and quick properties, but still we had 4,752 drug-like compounds. That's still too much to test, or too many to test. And then I went on essentially using Canvas and also some filters in CAT software to narrow this down to 200 compounds, which were then tested. And luckily there were a couple of active compounds then in the assay, and so that was then quite successful screening campaign. So it's possible in principle also for carbohydrate binding proteins. Heparinase is a heparinase processing, heparin processing drug, enzyme. And so it's possible to do that, but despite this, typically carbohydrates, carbohydrate binding proteins, which are either lectins or enzymes, are very specific for carbohydrates. So it would be essentially the aim to keep the carbohydrate that makes the specificity in the molecule and just to change the parts that are not interacting specifically in order to increase affinity. So it would be, instead of screening 5.2 million or even many more million in inverted screening, it would be nice to have a carbohydrate library essentially of compounds. So I wondered essentially how many carbohydrate containing compounds are in commercially available vendor catalogs. So the biggest one are Aldrich Market Select and e-molecules. So what I did, I took e-molecules and about 7 million compounds at that time. Almost 500,000 had stereo information, which is very important. Many of the compounds there have no stereo information. So even if they're carbohydrates, they are all like the same. So glucose, galactose are the same in these catalogs. It's not possible to distinguish between them, but if their stereo information is available, I can convert them into 3D and then use the CAT to detect the carbohydrates automatically. And this is what some examples that I found. And surprisingly, there are more than 12,000 carbohydrate compounds already commercially available in e-molecules, which is just out of the already one of the largest databases in the carbohydrate field. And so this is very exciting, but this is ongoing work, so we will see how that, whether this will be available at some stage as some library that we can use in the docking. When it comes then to another success story in the glycomimetics field, one is definitely Rivipansel or GMI 1070. Which is an E-selectin inhibitor, and it is further developed and also commercialized by a company called Glycomimetics. Development was done in Basel and in US, Basel, Beat Ernst and John Manani at Glycomimetics and other people, of course. So this is one of the recent success stories. Pfizer is involved in this now, so this is very excited in the field. If Big Pharma is going to license like carbohydrate drugs, and the other approach essentially is then to since carbohydrates have a very weak binder, typically millimolar or around millimolar naturally. So the one strategy is, okay, you have many of them interacting with a target, and this is very often used in viruses and also in other biological contexts. So you can mimic that kind of multivalency by making such kind of branched structures which have the active molecule at the end. And so here's an example that actually works against Shiga toxin, which is done in 2000, I think, 2000. It's called Starfish, and that neutralizes Shiga-like toxins. That's another success story. So this topic, glycomimetics versus multivalent glycoconjugates, is nicely discussed in that review down here of that group. and it's published last year in Chemical Reviews. Very interesting for you to read. I guess you get all the information essentially from the movie later, so don't need to write it down. Here is a structure of GMI 1070. As you can see, this molecule is not yet no more really like the Relenza case, where you have one carbohydrate ring and then decorated with some substituents that increase the affinity. So here we have actually quite a number of ring systems, and 3 of them are still kind of carbohydrate mimicking, and then another one functional groups, and here's some long linker. So I would be very curious to see that compound docked into E-selectin, but unfortunately there's no crystal structure available, but what is available is a substructure, and you can see here, this is how it binds. It's interacting the calcium ion here through the fucose, and that's what's originally sialic acid here is then mimicked by a very reduced scaffold that maintained the carboxylate group here for the interaction, and has changed the highly polar sialic acid against a hydrophobic scaffold, which makes favorable interactions in that area. What is kept is the middle ring, essentially, which is very important for having also specific interactions with the surface. Another recent very exciting example is TD139 from Biotech, which is a galectin-3 inhibitor. And Bristol Myers Squibb is now into that, and offered Galecto Biotech quite a significant amount of money for licensing in their compound. And here you can see this is still a rather large molecule, but it's more like what we probably consider as more like a drug-like molecule in the chemoinformatics. And recently a crystal structure appeared where that compound is docked into galectin-1. And this shows the profile, and this is very encouraging that actually you can get for lectin very low, well, middle nanomolar inhibitors, which is very encouraging. And so it's possible essentially. For a long time it was not believed that the lectins, which are typically have a very shallow pocket, are druggable. Obviously this is not the case, so they can be done. And the 2 examples that I showed last is actually quite some exciting examples that it's possible. The one is the Rivipansel, and the other one is this TD139. So as a general, when you want to do a glycomimetics design, so the key question you would be interested in essentially how does your ligand interact with the protein. Could be either the carbohydrate itself, or already then a carbohydrate that is modified in some areas where some parts of the carbohydrates are replaced by an organic substituent. And what we typically hope is then at some stage we would get a crystal structure, and this is also helps the modelers to start from a very solid starting point. If we don't have a crystal structure available of the particularly ligand, then computational methods actually might help to reveal the binding mode, and particularly here now the docking methods. As you can see here, this is a galectin-3 structure, and where the ligand is redocked into the binding site. I can see it works quite nicely here in the galactose area, which is the key interaction point here. That one is also very well, and the planar substituents, well, they are kind of flipped over. This is typically what the docking software cannot predict, because at the end of the day it's a planar ring. It could probably be both sides, unless there are some interactions in that area over here that are making specific contacts. So probably both versions are probably equally well in the score. Docking is all about post-generation and scoring. You should realize that if a pose has not been generated, that a certain pose of the many possibilities has not been found or tested, it cannot be scored. So if a very complex molecule, and carbohydrates are typically very complex molecules with a lot of rotatable bonds. It actually takes quite a long time to explore all the possibilities here and test them all against your potential interactions in the binding site. So it can mean that you're docking, you should probably use many more exhaustive sampling algorithms in carbohydrate docking than you probably need to use in the small molecule area, which typically only have a couple of rotatable bonds. So there are several methods that actually have used in searching of conformational space. Typical use in docking is stochastic search. FlexX has a slightly different approach. It's a build-up method. If a pose has been generated, it will be scored by a scoring function, which is independent, and hopefully the scoring function tells us then essentially, yes, this is a good score, and then we keep it. And at the end, when probably a post-scoring takes place, then we get the right order of the binding poses as a result. And that ideally the one with the highest score should be the one that is correct. So what I did here was a docking of a small ligand, a disaccharide, into galectin-1. And you can see on the left side, this is the sampled data of docking into about 17 galectin crystal structures. So each of them is very similar, but in terms of the docking, only half of an angstrom might make a big difference. So the good news here is that the poses with the lowest score actually have the lowest RMSD, and so this is how one would like to have it. The top ranked poses are the correct poses. When it comes to if we separate RMSD between the two residues, the one that should be, of course, very accurately reproduced is the galactose pose, which is this one. So all these ones that have the galactose in a good position, but some of them do not have the glucnag, which is the one that is at the reducing end, is a little bit distorted from the crystal structure. But overall, the blue one is the reference ligand in the crystal structure. So overall, here FlexX did a very good job. As a next test, I used TD139 and docked it into galectin-1 as well. And you can see now the situation is not as good as before. Essentially, there is none of the poses in none of the targets as the crystal structure produced, which is of course a little bit disappointing, but luckily there is a possibility to fix the problem. In this case, the key is this. FlexX offers the possibility to use a reference template, and we can have this command map ref in the batch script that runs the FlexX docking. And by using the galactose as a template, and we know in the galectin this galactose sits in this position, and if this has to be essentially this kind of constraint that we can use. So if we have that template, and then FlexX will try to put all the poses with that template satisfied, and then optimizes the rest of it. And you can see that there's a reasonable agreement with crystal structures. Again, here the planar rings are flipped, but the overall is quite a satisfying result. From the poll, I know that many of you are computational chemists, so you're well aware that the scoring is a difficult field, and unfortunately there's not yet a scoring function that satisfies our needs and allows us to blindly trust in the results of the docking. But there's possibilities to improve that a little bit in terms of post-scoring methods. One of the methods that was developed in our company here now is using support vector machines. Tanya Copsetti has used NIME and different descriptors derived from docking scores and different kind of measures in Schrödinger software and other software in order to derive a model for score prediction. And this gives quite exciting results, and you can see the correlation is quite nice. And it's also possible from that model that you can derive enthalpy and entropy components of the protein ligand complexes. Another method to improve the binding affinity prediction accuracy is use molecular dynamics simulation-based methods, particularly here expanded ensemble molecular dynamics with a double decoupling scheme. What you do is you have the system where the complex is built, that you need to have that as a starting point. Then what you do then, you decouple the ligand and from that complex the water flows in, and in a parallel window you couple then the ligand into the solvent. So what you sample essentially is a cycle. That's why it's called double decoupling. Here, because you decouple the protein and the ligand from the ligand from the protein complex, and you couple essentially the ligand into the solvent, and from that you can very accurately predict the binding affinities of carbohydrates to about 0.5 kilocalories per mole, and much more reliable than docking software. For example, docking, although some results here are quite good, but here, for example, Autodoc score would predict that the monosaccharide has a better score than the disaccharide, which is simply not true. It's the other way around. But the EMD method did that reasonably well. The same also MMGBSA is not too bad, slightly worse. But the computational costs are enormous. So it's about 25,000 CPU hours per value. So it's not yet completely for routine calculations, but the computer is getting quicker, and maybe the code could be improved. So maybe for the future it would be a very interesting method to use. General question is it possible to predict carbohydrate binding sites? Here's again a neuraminidase. It's not a monomer. Typically we would just link to the binding site. What I did here is look into all 3 tetramers. I did a docking, and you get the whole surface essentially screened. This is what you get, about 1,000 poses. And then the question is what to do with it. So I developed a method to calculate an affinity grid from the docking score. And if you go that grid essentially to higher values, then you directly end up in the binding sites, and it worked very nicely. The prediction, all 4 sites are predicted, and the method is described in that article, book chapter that is listed on the bottom. I did the same thing essentially now for not so specified target. Actually, it was a homology model of riproximine, which is a toxin. Which can hopefully be applied against cancer. It's related to ricin, so build a homology model. And since homology models are not completely reliable in terms of side chain predictions, I ran an ND simulation and then docked into about 1,000 snapshots. Blind docking. That docking took about probably half a year. But at the end it worked, and so the galactose binding sites are predicted, and so that means this one has probably similar features than the ricin, but there are a few more sites predicted which could be validated then by site-directed mutagenesis. So in summary, carbohydrates are difficult to dock because they have many rotatable bonds. The binding free energy is typically not very accurately predicted. The error is about 1.5 kilocalories, even if you train your set. It's possible to predict carbohydrate binding sites even on large protein surfaces, but you always have to face that there are also false binding sites available, but typically the ones that are correct are within the set, so experimental validation is required. And the blind docking even to fully flexible receptor is also possible, but that's also a huge amount of computational costs involved in terms of CPU hour, not so much about money, but kind of calculation time. So finally, I would like to then show you a design project which I was involved myself, so in collaboration with Reinhard Prosmer. Reinhard Prosmer, who turned about 90 this year, is a remarkable person. He's a synthetic chemist, a specialist in the synthesis of sialic acid derivatives. And so he is in Heidelberg and still actively in the office at university every day. So he founded a small company when he was about 80, and so what we did was then to aim at designing an inhibitor for cyclic 7, and Horst Brescher was also involved. He's a synthetic chemist, and Stefan Büttgemann did the biological testing, so a team of 4 worked on that project. Cyclics are mainly involved in the immune system, and particularly cyclic 7 is an inhibitory receptor expressed on natural killer cells. So it would be very interesting to find out what these molecules do in the body. For that reason, it would be also helpful to have a chemical knockout in the form of an inhibitor. And also at some stage probably could help in cancer treatment. And but cyclic 7 is probably the modeler's most worst nightmare. First of all, it's a lectin, that means shallow binding site. You can see here the sialic acid is essentially binding on a flat surface. Second, although there are a couple of crystal structures available of cyclic 7, each of them is a little bit different. So there are actually cavities formed, and so they are present in one crystal structure which are not present in the other crystal structure. So we have dynamic cavities, and yeah, that makes it not very straightforward. So the starting point then was to have select a few examples that covered certain dynamics essentially of the cyclic 7, and try then to dock or design compounds that would increase affinity by maintaining the binding pose of the sialic acid, which is important for specificity. Because of established chemistry, the first goal was to explore the 9 position, which is out here, and in order to find small organic substituents that actually dock into that cavity and hopefully improving activity. So as I mentioned already, the chemistry about the 9 position was well established in the Prosmer's lab, and also there were some successful examples already in other related cyclics where this kind of modification has resulted in improved activity. The best one at that stage was this one here, which is unfortunately only a 3-fold greater inhibitory activity than mu5Ac, which is the natural one. But okay, this just shows you many documents later. Finally, we came then to a compound that is 9N-oxamido compound, which is a picture shown here. It forms a couple of hydrogen bonds and also some aromatic interaction, which is always very important for getting a reasonable interaction profile of a small molecule. And what you can see here, it's about a 58-fold increase in activity in relation to the natural monosaccharide. So that was an improvement. So in that context, we looked at this 9 biphenyl substituted compound here, and the aim was to fill the 9 cavity with that hydrophobic moiety here. So in those days I used Autodoc 3, so the project is about 5 years, was performed about 5 years ago, and I got docking poses that actually dock into that cavity, but I also got docking poses that actually where the sialic acid is flipped over, and this is of course not very nice because the sialic acid is not maintaining its binding interactions. But what I realized at that stage essentially is this 9 position is exactly at the position where normally the 2 position in sialic acid is. So by changing the substituent from the 9 to the 2 position, and I was able then to generate a compound that actually have that biphenyl sitting in that tryptophan cavity here, which is in one of the crystal structures clearly present. And so I tested a few different types of linkers, and the chemist then, for synthetical reasons, went in for a triazole linker. This is the synthesis, how they performed. And I did then the docking, and also that compound equally well found this goes quite as predicted. And the question is now, of course, we can do a lot of modeling, generating nice pictures. The question is, what does the biology tell? And Stefan Wittgemann at the University of Heidelberg did then the biological testing. And as you can see, the light blue one, which is our new compound, clearly made a difference, and it's about 150-fold increase in affinity, which is a quite remarkable change, and of course we were all very happy that we got that. The compound was then further developed by biochemistry, and finally it has been then now in the 1 to 3 micromolar range, which is a very exciting outcome of the project, which probably took only a year or so. This is particularly because the chemists were just amazing, so they synthesized essentially these compounds like a factory. When you work in modeling, and this actually partly was actually one chemist really, because Horst Brescher did all the synthetic work, and this is just a great job. As a modeler, this is what you can always wish for, that the chemists are so quick that they come back essentially with compounds that can be tested, that goes into your modeling. So we could probably in 1 year go through 6 or 7 rounds of modeling, and it was very exciting to sit with these people in front of the computer screen and develop new ideas. And this is how it should work, interactions with modelers and computation and synthetic chemists essentially sitting next to each other in front of a computer screen. And then it can be very efficient, as it can be seen in this example. So, to our knowledge, this has been the design of the first cyclic inhibitor with a high affinity and a functional effect on cytotoxicity of NK cells. Interestingly, or very exciting for us as computational chemists, of course, that computational chemistry could contribute significantly at the beginning, but then efficient chemical synthesis was really equally important to go to the last bit of high affinity exploration. And at the moment, we do not have a confirmed structure, so only the model structure is there. It would be, of course, nice to have a crystal structure or NMR structure of the compound, but this is at the moment the current stage. So I show this list of applications here, mainly for the video later, so you can have a look. Essentially, there are a couple of interesting papers that you might be interested to read if you're interested in this topic. And with this, I would like to acknowledge my co-workers in the Cyclic 7 project, Reinhard Prosmer, who was very enthusiastic about the project. It was a pleasure to work with him. Horst Brescher, also particular pleasure. Stefan Wittgemann was also very important for the project. And the Heparinase project was also very exciting. Israel Ludavsky was always very interested in the project, and it was fun to work with, also Yinping Li. And finally, I would like to thank Per-Georg Nyholm. That's my boss here at Biognos. And as you can see, the Swedish West Coast is also a very nice place to be, and also in winter it has some nice moments, although it's pretty cold, and yeah, there's only a few moments light, so in winter it gets very dark here, but in summer there's essentially beautiful weather and long days, so you're more than welcome to visit Sweden at some stage. With this, I would like to thank you for your attention, and I hope it was helpful for your projects. Yeah. Thank you very much.
Thanks very much, Martin, for this super interesting presentation and sharing all these insights into project work.